You've probably seen a zebrafish before, right? Maybe in a pet store or an aquarium. But this common, everyday little fish is hiding a huge genetic secret. And it's a secret that it's totally changing how we understand our own health and diseases. So, let's dive in.
So what's the big deal? What exactly makes this tiny fish not just a pet, but an absolute powerhouse for biomedical research? Well, it all starts with just how much we have in common. Get this, 70%. That's the percentage of human protein-coding genes that have a counterpart, an evolutionary equivalent in the zebrafish. And that includes most of the genes we know are tied to human diseases. It's a pretty incredible genetic overlap. Now, that genetic similarity is amazing for sure, but it's not the whole story. The zebrafish's real secret, the thing that makes it such a game changer for science, is something much, much weirder and more powerful about its DNA. Here it is, the big secret. The zebrafish genome is absolutely packed with duplicated genes. I mean, extra copies, redundancies everywhere. So where we usually have just one copy of a gene, the zebrafish often has two. And that little quirk? Well, it turns out to be a scientific goldmine. So you have to be asking, where did all these extra genes come from? To answer that, we need to take a trip way back in evolutionary time to a single, massive genetic event that completely reshaped an entire branch of the tree of life. Okay, so here's what happened. All vertebrates, that includes us, are the result of two big ancient rounds of whole genome duplication, way back around 600 million years ago. But the ancestors of bony fish, like our friend the zebrafish, they went through a third round about 350 million years ago. This essentially gave them a complete backup copy of their entire genetic library. This is called a whole genome duplication, or WGD. And you can think of it like this. Imagine your DNA is a library of instruction manuals. A WGD event is like someone just photocopied the entire library, every single page, and handed you a second complete set. All of a sudden, you have a spare copy you can experiment with. You can edit it, rewrite it, even rip pages out, all without messing up the original. It's the ultimate raw material for evolution.
So this, of course, leads to the billion-dollar question. What does evolution actually do with all these extra genes? For 350 million years, these duplicates have been on their own evolutionary paths. Let's take a look at what happened to them.
It really boils down to three main possibilities, three fates for these duplicated genes. You can either lose it, split it, or change it. Pretty simple, right? Okay, so the most common thing that happens is just lose it. One of the two copies just collects mutations over time until it stops working. It becomes a pseudogene, which is kind of like a genetic fossil. It's the easiest way for things to go back to the way they were. But things get way more interesting when both copies stick around. And the most common reason for that is split it, or what scientists call subfunctionalization. Basically, the two new copies divide the jobs of the original ancestor gene. It's like a single multi-tool being replaced by two separate tools, like a screwdriver and a pair of pliers. Each one is simpler, but more specialized. The perfect example is the gene PAX6. In humans, this one gene is a master multitasker. It's got important jobs in the eye, the brain, and the pancreas. But in zebrafish, the job is split. There's PAX6A and PAX6B, and they've divided the work. And this is huge for scientists because it lets them study the gene's role in something like diabetes completely separately from its role in neurological disorders. That's something that's way harder to do in mammals. And that brings us to the third, and maybe the most exciting fate of all, change it, or neofunctionalization. This is when one of the gene copies is freed up from its old job, so it can evolve a completely new one. This is how evolution builds brand new things. For instance, check out the HOXA13A gene. It started as a duplicate, but it started changing really, really fast. While its twin kept doing some of the old jobs, HOXA13A evolved a totally new function, building a special fin structure. This is a crystal clear example of how gene duplication can literally create new body parts. Okay, so this isn't just a cool history lesson about evolution. This unique genetic setup in zebrafish gives us an unbelievable blueprint for untangling the complexities of human disease. There's this quote from the source material that just says it all. Each pair of these duplicated genes represents a 350 million year long natural experiment. Trying to recreate something like that in a lab would be basically impossible. If we only look at one of the copies, we're missing half the data. And just look at what this experiment shows us.
For the gene SCN1A, which is tied to epilepsy, the two zebrafish copies help researchers tease apart different neurological functions. For SOX9, they help us understand how one gene can control both face and ear formation. And for TBX5, they let scientists figure out the specific jobs of different parts of the protein involved in heart defects. It's like having a detailed instruction manual with all the subsections neatly separated.
This principle is also incredibly useful for modeling complex diseases. In some breast cancers, the HER2 gene gets amplified, meaning the cancer cells make extra copies. While the zebrafish's naturally duplicated genes give us a perfect model to study that. It even helps us understand how cancer cells become resistant to chemotherapy, because they often do it by, you guessed it, making extra copies of certain genes.
So it all comes back to this one, really simple but powerful idea. You have to look at both copies. The real magic, the deep insights, come from making that comparison.
And that means the zebrafish is not just some convenient stand-in for a human. It's so much more than that. Because of its unique evolutionary past, its duplicated genome gives us a special window into how genes work, a way of deconstructing complexity that we literally cannot get anywhere else.
So, let's wrap it up with the key takeaways. The zebrafish genome was totally transformed by a massive gene duplication event millions of years ago. This set up a huge natural experiment that split the complex jobs of single genes into simpler, separate parts. And by studying these gene pairs, we get this incredible detailed look at the very building blocks of our own health and disease.
Which really just leaves you with one last thought. All of this incredible insight comes from a tiny little fish you can find in any pet store. It really makes you wonder, doesn't it? What other profound secrets are out there, swimming, flying, or crawling all around us, just waiting for us to take a closer look?